Right. Hello, everyone. Thank you for joining this presentation on the MERSCOPE Visualizer. So I'm Sayak. I'm a senior field application scientist with Vizgen, and today I'll be talking to you about the MERSCOPE Visualizer software and how it helps the MERSCOPE user realize their data once they capture it from the system. Um, Before I begin a demonstration of the visualizer, I'd like to mention that the visualizer software comes free of any licenses, which means that once you have MERSCOPE or MERSCOPE data and you want to look at the data, the visualizer is made available to you both in the Windows and the Macintosh environments that you can install on your computers and open the MERSCOPE visualizer related data on your systems. So this is not locked by any licenses. We just ask you register your username and register the installation on your computer, and you are pretty much good to go. The other thing I wanted to mention is that you may have noticed already that your microphones are on mute, which doesn't stop you from asking questions because there is a Q&A button on the top of the window, which you can click on and ask questions. I will try my very best to answer your questions during at the end of the my demonstration. However, if I fail to answer questions due to lack of time, or if those questions need a little bit more research and help from our very knowledgeable and highly skilled bioinformatics team, we'll definitely reach out to you and we can talk offline. My email is provided here below at shayak.mitter@vizgen.com. On the screen, on the left of the screen, you see the MERSCOPE, the key components of the MERSCOPE hardware. But I wanted to talk about the data output that comes from the MERSCOPE for each run. Now, the MERSCOPE solution is compatible with fresh frozen FFPE tissue types. So there is a lot of versatility there. Plus, it is also compatible with cells, which means both fixed and fixed as in adherent cells or suspended cells. Whatever be the sample that you are scanning on the MERSCOPE, you are able to draw multiple regions of interest depending on how much of that area in X and Y you want to capture. For every region of interest that you capture on the MERSCOPE, the data output consists of these files that you see on the left here. So right from the top, you get the list of detected transcripts that comes out in a CSV file. This will contain the X, Y, as well as Z information of the transcripts that are being detected by the MERSCOPE algorithm. And X and Y as in, in single plane, but because the MERSCOPE is able to do multiple Z planes like another microscope, you are able to get that three-dimensional data as well. Next, the mosaic images are these high-resolution TIFF images that consist of different stains that we use. So for every MERSCOPE run, we use DAPI and poly-T. DAPI, as we all know, stains the nuclei, whereas poly-T is a stain that targets the diffused RNA in the cell, which is more of the nuclear and cytoplasmic marker. In addition to that, you are able to use cell boundary stains that Vizgen provides that help with downstream cell segmentation. This comes as a part of the three targeted cell boundary staining kit. Plus, you can also do protein code detection up to 6 different proteins of your interest. So all of these different stains, that is, once again, to recapitulate, DAPI, poly-T, cell boundary stains, protein stains, each of these images the stains are captured as like a microscopy image, and they are stitched together into this very high resolution mosaic images and supplied to you as TIFFs for every Z plane. Next, transcripts per cell matrix. This is the single cell data that you get for the out of the MERSCOPE, you get MERFISH analysis, plus you get cell segmentation. The cell each cell identity with its corresponding gene expression matrix in terms of copy numbers of genes detected within that cell entity is expressed in and shown in this transcripts per cell matrix. The cell metadata consists of the spatial information of the cells. So for every cell that has been identified, we provide you with XYZ coordinates, as well as with an average diameter, the volumetric information. Furthermore, any kind of protein stains that you have used or the cell boundary stains, they are the sum intensity of each of those stains are also supplied in this cell metadata. So it gives you semi-quantitative information about your protein targets. Finally, there is the cell boundaries. This is available in a Parquet file format, and this consists of the cell segmentation mask information that is derived from the cell segmentation algorithms. Now, as you can already start to see, that these are very commonly used file formats, and they are, in fact, all portable into commonly used single-cell analysis pipelines such as Seurat, ScanPy, or the more newly established methods such as SquidPy for spatial data. So it's very open in terms of what data it's providing you. However, to help the biologist quickly visualize the data that they have captured from the MERSCOPE system, we provide this additional tool called the MERSCOPE Visualizer. The MERSCOPE Visualizer encompasses all of the information that you see A through E on your left and makes it a separate additional file for every region of interest that you scan. And that can be opened on this software, and you can start to visualize data. I do want to further mention one small additional tool that we provide to help our users. And this is the Vizgen post-processing tool. This post-processing tool helps with segmentation of data on your helps with cell segmentation when you're if you the cell segmentation that you have acquired from the MERSCOPE is if you feel that there is room for improvement, the Vizgen post-processing tool helps you with identifying cell segmentation. So this is an additional tool that we provide. With that, that's just a single slide that I wanted to show you for the quick introduction of the MERSCOPE data output and the visualizer introduction. So here are the different links that you have to see here. So any kind of queries about the MERSCOPE product itself or the visualizer, please email to info@vizgen.com. Also, definitely visit our website for more resources. Now, the data set that I want to show today is a human breast cancer FFPE dataset. This is the MERSCOPE visualizer that you are looking at the moment. Before I jump into the visualizer, I do want to mention that this dataset is also available as a part of Vizgen's FFPE data showcase. So I strongly encourage those of you who don't know about this yet, definitely visit Vizgen.com and go and look under the resources under the data release program. And here are the different data releases that we have. We are currently looking at, on the other tab, this FFP human immuno-oncology data, and there are multiple data sets here, starting from breast down to uterus, colon, ovarian, etc., on multiple patients. And this is an interactive web visualizer, and it gives you a taste of what the visualizer, the local installation of the visualizer is. So you can definitely play with the data online yourselves after signing up for access, and you can actually download the different files that the CSV files that generate, the CSV and the TIFF files, etc., that are generated from the MERSCOPE. Right. Let's jump into the MERSCOPE visualizer itself. So when we open up the MERSCOPE visualizer, we have these different display windows. This is the GUI for the visualizer. Right on the top left, we have the File, Edit, View, and Help menus. I commonly find that the File menu is perhaps the most useful for me. So here, through the File menu, you can open a saved experiment that was saved as a saved visualizer experiment. You can load recently compiled visualizer experiments through this list here. You can import different types of data from into the visualizer from your bioinformatic analysis pipelines. The visualizer plays extremely well with ScanPy scripts, which are commonly used in bioinformatic analysis of single cell data. So this is where you can import data in the form of AND data, CSV, or import geometries, and then overlay onto your spatial data. Then there is the exporting of data as well. So the visualizer does let you export out annotated data in different formats as well, in terms of CSV and geometries. So this is perhaps most useful of the four different tabs. With the edit, you can definitely look at and manage the imaging channels, which are the different staining channels. And the view is these are the different the little windows, which we'll talk about in a second, such as the scatter plot, spatial features, etc., that are currently open. So when they're checked, which means that they are open at the moment. And furthermore, we have the help where you can find out about the Vizgen, or you can open the visualizer log folders through the report folder. Right, that is enough about that little menu bar over there. Now, that perhaps the biggest window that is looking at us right now is the spatial window. So this is where your tissue or your cell sample, whatever region of interest you have, is shown here. However, when you first open the visualizer on your computer and you're about to load a data set, obviously there has to be a workflow for importing that. This is very well described in the visualizer user guide. So we do have a very detailed MERSCOPE Visualizer user guide that you may refer to when you're handling this. But I'm going to quickly show how to open or assemble experiment, as we call it. So we access through the file. You go to this assemble experiment. If I left click on it, this opens up a pop-up menu where you can write down your experiment name, whatever be that experiment name. You select an output folder where you are going to save the visualizer experiment by selecting this folder and then selecting the path where this is going to. I, for example, have it on my D drive as MERSCOPE data under MERSCOPE data. And then what you have to do is select the files that need to be assembled to create this visualizer experiment. And the file is in a format called a .vzg. The .vzg file gets generated for every region of interest the MERSCOPE scans, and you can assemble those data sets through this function here. So you click on the folder and you go to the respective path wherever your VZG files are, and you can double click on that or click on it and hit open, and it gets loaded onto the list here. Now, you may have multiple regions of interest. You may have multiple experiments, all done with the same probe set or the gene panel. In that case, you will again click on the select the data set and direct it to whatever other folder there is another VZG file, and you can again with the same way select open, and you can populate this on the list. Once you have incorporated and put in all the VZG files, you then click on the little pink button on the bottom right of this assemble experiment dialog box, and you are basically then able to assemble the experiment. The assemble experiment does take about a minute to maybe 5 minutes, depending on the number of data sets that you're looking at and the size of the data set in terms of the area of the tissue as well as the size of the gene panel. So for time's sake of time, I'm not going to build the experiment over again. I've already done that work and I've saved it, and I've opened this as through my file recent, and I've had this saved as this human breast FFP under MERSCOPE data, and I basically clicked on that and this has opened up. Now, So that is all I had to say about the assemble and operation on the MERSCOPE Visualizer. Next, let's talk about the spatial window in the center. Now, right on the top of the spatial window center, or spatial window, you do have a few functions here, of which you can obviously start to guess what these are. So transcripts, I'm going to turn off all of them. So this is now a clean slate. And then you have images. So if I now click on the images, these are the microscopy images that are being output by the system. So these are several thousands of field of view, and the area of this breast cancer tissue is about 80 square millimeters. So this entire tissue has been stitched together into this high resolution mosaic, and you can then zoom into it using the scroll wheel of my mouse. So that's what I'm doing. I find this to be the most useful. However, there are two other options to zoom into this. So I'm going to quickly show those two options. So first one is the able to use the little window here, the buttons here. So if you hit subtract, and you can go, obviously you can zoom out, plus you zoom in. Furthermore, there's a drop-down menu if you really want to quickly zoom in or out, you, depending on like 100 microns, or if you want to go out into this, you are able to quickly select different zoom factors to look at your tissue to sample. So while you have the images of the different cells that of your tissue and the different stains, so then you can turn on something like the cells, for example, and now you have the cell segmentation being shown around. So I can left click, drag this, and move this around to look at my sample. Now, if you are really zoomed into your sample this much, and then you want to quickly navigate from one part of the tissue sample size to the other, then you do not have to like drag and move it around or zoom out necessarily. What you could do is click on this little mini map on the left here, where you can see the entire thumbnail showing the as it says, the mini map of your tissue, and there is a little red square, which is that is basically where we are at the moment. So I'm going to left click and drag it, and I can quickly move to another part of the tissue for quick navigation. So I would like to turn that off for now, so I can left click on it and turn it off. Furthermore, if I want to see what data set this is I'm currently looking at, because you may be able looking at multiple data sets, unlike today's demonstration, you may be actually loading multiple data sets. In that case, you might want to know which data set you're looking at. So if you click on this little information button, you are then able to see what data set this is, and what is the window size currently, and etc. Any view that you have, whether that be images, cells, or if you put transcripts on top of it, any view that you have in this entire window can be quickly exported out as a high resolution image for whether that be required for publication or for your lab meeting presentation or for a poster. So here on the right, there's this button here, which is the export PNG button. If I click on this, everything that I have here will be exported out to whatever address that you want to export it out to. And then once you click save, it also lets you select what resolution factor you can select. So you can, there's the wide range for selection. So if the higher you go in the resolution factor, obviously the higher your resolution will be. So here I've actually I'm going to try and open this if my computer would allow me to. Trying to quickly show you a couple of these regions that I actually have. Just this one region which I kind of exported out. So here are the microscopy images. I have the eye information on for this tissue, and it's showing the different stains and etc. Then overlaid with cell segmentation, and finally overlaid with the images of the transcripts in that region. So that's the little quick overview of what these top buttons do. Now, these are there because these are typically the first things that you might want to look at. You are going to definitely need transcripts, cells, and images. But then there are finer controls, and those finer controls are hidden under the settings toolbox in this spatial settings control. This is a little wrench here on the right, and I'm clicking on it, and now it really opens up the different aspects of and the controls associated therewith. So on the top, you have the data set. So if I click on this little drop down, obviously I only have one VZG file open. If you have multiple VZGs, you will see a list of the VZGs here, and you can click on one of these and any one of them, and that will be displayed out here. Now, just want to quickly escape from the spatial settings button because I do want to show you that there is a two-window option here. So in case you do have multiple data sets, you are able to access this second window, and it has its own settings, and through the spatial settings, you can actually pull in a second VZG file. Here, it's somewhat silly that I am going to use the same data set, but to show you, but you can basically look at the same data set or different data sets side by side, whatever displays you choose. So I think that really allows the user with some convenience in visualizing their data, and when they're particularly when they're looking at donor versus control, etc., they are able to visualize things quickly. Let's get back to more of the controls here. So I mentioned right in the beginning that the MERSCOPE can do multiple Z-planes. So here, as I zoom in, you will be able to now see that these are right now, only the DAPI and the poly-T stains are being shown. So DAPI is shown in blue, whereas poly-T is shown in green. So now this is on plane number 3. So if I go to plane 0, which is closest to the imaging slide, then you can see how the microscopy images are changing as I toggle through the different Z-planes. And I move to the other extreme, which is the 7th plane that is farthest away from the objective. I'll go back to plane 3 because that's about the center of the sample in terms of Z. Now, you can then look at the other layers. So we have transcripts. So you can turn on the transcripts. And if I zoom into the transcripts, you can start to see those little dots that are the gene transcripts that have been identified. If you hover your mouse over those transcripts, you are then able to see in that drop-down balloon next to your cursor, which shows you what is the name of the gene. That is, like, for example, I'm looking at a gene that's named PKM. It's at this XY center, and the transcript count. That is for that particular gene that has been, how many times it has been identified in this sample. Now, even those can be visualized per plane. So so here it says Z plane active. So if I click on this drop-down menu, there is another option which I'll come to in a minute, but I can now start to change my planes again. And again, you'll see how these transcript distributions in each plane is being displayed on the visualizer. Now, for example, if you are not really interested in looking at per plane information and you just want to know that over, say, a 5-micron FFP slice, how many transcripts are there in the entire Z, all you have to do is, instead of having active plane, which active refers to the plane that is being highlighted here, you have to select all, and you then get to see all the different genes that are being expressed in this entire scene. So what it is basically an axial projection. So it takes all the 7 optical sections that have been scanned in this data set and squishes them somewhat together and creates an axial projection data. So now you can start to visualize everything that was detected in that entire through that Z plane. The next thing is the cells. So here now you can see how the cell segmentation is being applied. And then again, the cells can be also visualized over multiple planes with the active and all button. So you do see a couple of these sliders here. So these sliders are basically controlling the transparency or the fill color transparency depending on what targets they are. So for the transcripts, if I just turn on cells for a second, right now they're completely opaque. You can control the opacity and transparency. This might become useful, particularly when you are overlaying different things like images, cells, transcripts, etc. It might become really, really useful to have that function. Furthermore, for cell segmentation, it initially opens up and it's a very opaque, like with strong colors. I typically like to leave it somewhere down at the 25% opacity, so I can actually see what cells are within, and I can be able to see the images of the microscopy images of the DAPI, poly-T, etc., which I hope you can see as well, that are underlaying and kind of be able to judge the cell segmentation quality. Right. So let's talk about the images. So I'm going to turn off the cells or transcripts either by clicking here or by clicking on top of the window. And now let's talk about this, the cells for a little bit. So I mentioned that this has DAPI and poly-T, but it also has a few other stains, which is the cell boundary stains that we see here. So very quickly, when I zoom in, I can turn on the poly-T. You can play with the lookup table. These are 8-bit displays, displayed as 8-bit images, which is why you see a 0 to 255. The visualizer is capable of displaying full dynamic range, as many of you who are MERSCOPE users may have already seen that some of this data is being expressed in a much higher dimension, dynamic range. But here, other than the DAPI and poly-T, let's also look at the cell boundary stains that we have used. The cell boundary staining kit is extremely useful in helping the deep learning segmentation that we now have incorporated on our platform. So what it does is, in a tightly packed tissue environment such as this, it will target. I'm going to turn the, actually before I display that, I'm going to turn the color of DAPI to grayscale to help us visualize this better. But I'm going to turn the DAPI off for a second and maybe push the lookup table a little bit to show you how the cell boundaries has been applied. So you can start to look at the cell boundary and then that with the DAPI, so you can start to see how the cell boundary stains have been applied. So this is an extremely useful target, and I strongly recommend people who are looking at samples that have packed tissue cells. The cell boundary stain kit from Vizgen is very good at staining and helping with downstream cell segmentation. So here for the breast tissue, we can see the cell boundary stain 3 as well as cell boundary stain 1, cell boundary stain 2, which may not have performed as good as cell boundary stain 3, but the other stain that has performed really well is cell boundary stain 1 in magenta. So this is when I'm turning on. You can play with the brightness, contrast a little bit as well. And once we apply the cell segmentation, this has been analyzed with cell pose right out of the MERSCOPE box. And you can start to, like, when you turn off the cells on or off, you can kind of start to see how the segmentation has performed. Right. So that is all the key information that I had to mention about the spatial settings. There are furthermore, there's one definite, couple of more useful additional settings that I want to mention here. One being the rotation of the image, as it mentions. So if suppose you want to have a rotated image, if you're particularly if you're looking at something like the mouse brain, and its sample is a little rotated, and you want to rotate it correctly before publication, so you may be able to just type like positive numbers for clockwise rotation or negative numbers for so all you have to do is type the number in and hit enter, and the image rotates. So this will hold when you again export the data out. It is going to that is going to be exported out in that orientation. So I'm going to turn it back to 0. The other one is the background color. Now this is really useful if you are publishing for for publication standards, you need a white background, so you can definitely change that background to white. This will work for both transcripts and cells. However, for images, just because they're microscopy images, we require that the background be black or darker backgrounds for better rendering of microscopy images. So that is those are the key things that I want to talk about, the spatial settings. Now, to talk about the different genes that are seeing here. So this on the left here is your gene list of genes that were in this gene panel that we use for probing this target. So right out of the when you open this right out of the box, this is in this big large list called ungrouped. So here, if I uncheck all the genes, obviously everything vanishes. But then if I start to highlight, click on check on the boxes and highlight, for example, now I have ANGPT2 showing up. And then you have, if I click on another button here and I highlight those two holding the control button down, I can start to highlight whichever genes I want, and I can look at them as, whether that be just by the transcripts themselves or in the spatial context with relative context with the cells. I can turn the cells on and look at them. So while this may be a useful way of navigating around the data, particularly when you have multiple data sets you're looking at, you might want to actually group the genes together so that you're quickly able to visualize, say, for example, some cytokines that you're looking at, or some other cell markers that you might be interested in. So the one way of doing that would be right through here, where you can actually check on different genes, and you can, on the bottom here, type out a new group and put them in a certain group, and you just hit save, and it'll get transported moved to a separate group, such that when you check that group on, only that group will be displayed here. That's one way of doing it. I find it much easier to actually do it sort of outside the software and export it and import it back into the software. So I'm going to quickly show that workflow here. So when I right click on this, you can actually export out the genes here. I have already exported it out, and I've named it the ungrouped CSV. So let's quickly look at where that is and how we can manipulate that. So I have it saved under this gene list. So if I double click and open this, here I have the different gene names. So they're all listed here in the color map based on whatever display colors are being used. And then everything right now says ungrouped. So I can start to go and type in a different name, such as whatever that be. I'm just using something like a highly expressed term, and once I save this, I can then import it back into the visualizer. So I will close this one, and then I do want to bring out, just open one that I'd already annotated. So here in this, I have actually saved it as this group, and I just use something. Obviously, you're going to have more biologically relevant annotations, and you can then import this back into the gene list. So if I right click and I hit import, and then I point it to that CSV file where I've made those changes in the names, double click on it. Now I have this separate gene where some of the genes from the ungroup that I wanted to be moved to a separate list, and now they are being grouped together as highly expressed, which means now that if I just have these checked, these are the different highly expressed genes such as BCL2, BRC5, BEST2, etc., that are being shown. Obviously, you might have low expression or something like cytokines. You're going to do more biologically relevant groupings. So this is just an example to show you how these genes are being, they can be grouped together. And I find this really useful, and you can save that and import that CSV file into multiple data sets as you go, as long as they have the same gene panel that was being used for interrogating the experiment. Right. So To recapitulate so far, we've looked at the spatial window, we've looked at the settings, we have now looked a little bit into the genes, spatial, how to look at the spatial spread of the genes. And then now, the next thing that I would like to show is the single cell clustering, helping with the visualizer. So the visualizer doesn't do single cell clustering within the software itself, but it plays very well with things such as pipelines such as ScanPy. So through the file export data format, you can actually export the annotated data, the AND data in an HDF5 file format. What happens then is that you are able to create this based on the entire data set. If you draw regions of interest, you could export it out based on a single region of interest or different regions of interest. You can export out the HDF5 file. So I have already exported it out and I have saved it under as this HDF5 file, as this file. So this file basically contains the cell, the entire cell gene expression matrix, and that file can then be ported into your ScanPy algorithm. So Vizgen's visualizer actually does provide the user with such a script. So here we have that script where you can actually load the HDF5 file. And if I run this ScanPy script, I will be able to first filter cells based on minimum gene expression counts, as well as them in volume, and then proceed to single cell clustering. This single cell clustering file can be accessed from within the Vizgen's MERSCOPE user guide, or it can be accessed through the MERSCOPE resources that are available on Vizgen's website through the visualizer clustering template. Once you have done the clustering, whether that be using this particular notebook, or if you have run it locally, depends on what your analysis pipeline is. As long as you have a HDF5 or H580 file formats, you are able to port it back into the software, and you are able through the import data format as an AND data. And then once you import it back, you are then able to visualize the single cell scatter, the cluster plot. So the cluster plot has to be in a UMAP or TISNI format. So here we have a UMAP. So now that cluster can be overlaid onto the cells here on top of the tissue. So now you can see the spatial distribution of the different cells in that scatter plot. So now you have a few more interesting things that you can do with the visualizer. So you can actually point to different cell clusters by just simply clicking on your UMAP scatter plot, and here all the cells that are part of that part of the scatter plot cluster are now being displayed here. Furthermore, on the top left, this is the spatial features window. You can start to see that the different cell clusters that are identified by your cell clustering are now being displayed here. So you can either toggle through here, just like your gene list, or you can hit the control button and select multiple and overlay them together. And you can see how those are being highlighted on your scatter plot. Now, just because we are in that talking about the spatial features and brought a little bit of that into the discussion. So let's talk a little bit about the spatial features window here. So the spatial features window does have a few interesting functionalities. So here, as you can see right now, we are showing the different cell clusters that were identified. So here we have the cell clustering was done using the Leiden. You can do it whatever kind of algorithm that you want to use, if not Leiden. So we have those Leiden clusters, and within the parentheses, we have the number of cells that were identified in each cluster. So now you can definitely color, using the spatial features, you can color your spatial tissue map, sample map with the active category, which is the Leiden. Or you could move to some other format, and I'll hopefully have some time to talk about this as well. But you can, whatever is the active category in this, that will be the color that will be applied. However, there are some other functions which are really, I think they're really cool. So for example, counts. So if I do that, then basically for the entire whatever genes that are right now selected here, the display map here in the spatial will tell us how, like, what's the expression, like which of the cells that have the highest counts. If I zoom in, you can see that some cells do tend to have higher counts than the others. So that's just basically raw counts of transcripts, how many transcripts are being counted. You can play with the brightness to further, bump up and how these play with the min and max to bump up how this is displayed. If you want a more standardized form, such as z-score, which is like how many standard deviations above the mean the expressions are fluctuating, whether above or below the mean, you can then display it based on z-score. Furthermore, you have normalized, which is basically normalizing based on the cell volume. So you can have that. So that is count, just the transcripts counts displayed by cell volume. And then you have the normalized z-score, which is again the z-score that is just normalized with the cell volumes. So you can display that as well. So these are some of the additional data that you can visualize from the visualizer. So now when you look at the active, go back to the active category, which is where I want to be able to show you some additional functionalities. Then you can start to look at some of the features of the heat map. So there is a heat map that is very useful in the visualizer, and that can be pulled up by left clicking and dragging out these buttons here. So, and I just keeping it here so that we can kind of also get to see what's on the data, spatial data here. So this is the heat map of the this data set based on the Leiden clusters. Whatever heat map you want to be displayed here, you can go under the heat map settings, which are, again, just like the spatial settings has a range, the heat map setting window also has a range to it. This can be clicked on, and you can basically click on whatever the active category is by grouping by category, and that will basically draw the heat map based on whatever active category has been selected under the spatial features window. So here we are now seeing the Leiden clustering. So right now we have the option of sorting and filtering this out. So now, right now, it is saying that genes have been sorted based on count, and the categories, which are these categories of the Leiden clusters, they have been sorted based on the counts. So basically, if I just make this go away by clicking on it, you can see that from left to right, these clusters have been arranged in such a way such as the cluster 4, 2, 3, and in descending order, they have the highest amount of transcripts detected just by counts. If you want to sort this based on something else, such as variance, you can perhaps do that. Then you can start to get a little bit more like z-score wise comparisons. Furthermore, if you want to create hierarchical, you can actually group these genes together amongst these clusters, and then you can have these dendrograms, if you will, that kind of tell you that basically are telling you how these clusters, these genes are being clustered together, then they have similar expression profile. You can actually play with the slider on top here when you have the display on hierarchical methods. So right now, if you have all the 550 genes, if you start to then sort of draw more of this clustering, you can start to see how the dendrograms are starting to draw out, and cluster these genes together. So these, for example, this cluster here seems to be very highly expressed in cluster 0, 3, 2 to 4, and you can then right click on that cluster, and that cluster, if I turn the transcripts on, those clusters will be highlighted on, the genes in that cluster will be highlighted on the list here. And you can also right click on this, copy genes, and put them on a list, and now you have basically all the genes that were highly expressed in clusters 0, 3, 2, and 4. Basically, you have export that information already. So this kind of information really helps, I think, with downstream bioinformatic analysis. So now if you wanted to take even a more zoomed view, so let's just look at a few of these top controls here. So the heat map does let you zoom in to the data set. So you can actually click on this particular one. So if you are really interested to see, okay, what is this particular gene? And you can then zoom in in that region, and kind of immediately zooms in and shows you what genes are being, the name of the genes. If you want to quickly bounce back to the original zoom, you can either use the scroll wheel, or you can simply click the reset zoom, and it goes back to where it was. From under the settings, heat map settings window, you can do a few more sorting. So while we were clustering the genes based on hierarchy or counts or variants, you can also cluster the categories based on their. So if I do hierarchical here, it's going to immediately place these clusters in a position where it feels that these are closely related. So again, clusters 0, 3, 2, 4 seem to have similar expressions patterns. If I click on that cell cluster, turn cells on, you can now start to see that these 4 clusters have been highlighted here on the spatial settings window, as well as on your tissue. So you can now see how these are. So this can really help start to realize some of those neighborhood analysis as well. Of course, you can play with the sliders as well to increase or reduce the clustering factor, and display the data accordingly. Now, whatever data I see here, I am able to export it out as a heat map. So here, for example, I do have, if you can see, I'm not going to try open it in the interest of time, but you can actually have this heat map exported for publication. Now, any cluster that is selected on the spatial settings, that can actually be exported out. So this, I would just want to kind of go into a little bit about the export, more into the export functions that are there in the visualizer. So you can actually right click on any single cell cluster, and you can hit export, and this is going to export out the entire cell index numbers that have been identified by cell segmentation out into a separate CSV file. So here on the right, you see under the active feature set, all the details of the identification details of those clusters are being changed and displayed as I toggle through each of these clusters. You can see them. So if you can export them out as I was saying, due to a CSV. Furthermore, if you want to create cell groups. So let's talk a little bit about the manual cell groups. So looking at cell clustering is great, but you may be wanting to, based on your tissue architecture, you might want to actually manually annotate groups and extract information from there. So for that, we actually have something called custom cell groups. So here I have actually drawn a few custom cell groups here. So I'm going to click on this so you can start to see these different polygons that I have drawn. So I'm going to just quickly show how that is done. So here is the rectangle selection and the polygonal lasso selection. So you can just simply left click, drag out, and actually I have to zoom in a little bit, and drag and draw, and then that group will be selected, and you can actually save that group. So in the interest of time, I have saved some of these groups, and that's why you could see those polygons. You also have this polygonal lasso feature, which is, I think, more biologically useful. You can create a group like that, and then at the bottom you have whatever cells have been identified. You left click here and you type out a name of the group. So using that workflow, I have already saved a few such groups. So here, if I just uncheck the unclustered for a second, so basically I have group 1, which is this little square box, group 2, which is actually a full cluster that I have saved, and and similarly, group 3, group A, B, whatever is it. And again, once again, just I mean, I know these are silly names, but you will probably have more biologically relevant names for tissue structure. But basically, I just want to demonstrate how this is done. So now, again, whatever active category is selected in the custom cell groups, you are now able to look through the heat map that again shows you the same the different groups here that are being displayed through. So you can now see that the different the hierarchy of the cell groups that are shown here, as well as the gene expression. So you can then start to again play with the thing as a group A, B, and perhaps 1 are similar based on where you want to leave that slider. And then again, you can start to see what these gene expression is. Right. Coming back to the heat map, and I apologize, I did kind of went back and forth between two different things, that is the spatial features and the heat map, trying to cover both cell group as well as light and clustering. But I just want to show this how seamlessly the heat map behaves with each whatever you have on the active category. So you can actually then start to look at a few more things. So for example, here you can actually use the visualizer to filter out genes based on the variants. So you may not be interested in looking at all the genes. And if you want, you can simply, for a second, just export out here. If you hover the mouse over the genes, you can actually use the mouse scroll wheel to zoom in and then start to display what genes are in each of these regions, and you can visualize that information. However, in addition to that, if you just wanted, the top 50 or top 20 genes, so you can actually use the slider to filter out a lot of this information and drop a lot of the genes from the display. So if I just keep it around, say, this 36.9 variants and the number of genes such as the 20, and you hit apply, and then you are basically able to then see only the top 20 genes. And of course, you can then play with their display a little bit, and based on wherever you want the expression, the coloring based on the expression, you can actually start to display that. So these are the key information that I wanted. I think I have about 5 minutes left. So I think there are a few questions that I see here on the chat. Please feel free to ask more questions. So as you zoom in, do the cells in the UMAP plot reconfigure? No. So the scatter plot UMAP, if I understood your question correctly, the scatter plot stays constant in its form here. So basically, it reconfigures to whatever active category you have here. But as you zoom into the cell, zoom into your sample, it doesn't need to reconfigure. You can pick whatever your sample is like. If you zoom in, the scatter plot stay the same. You can actually zoom into the scatter plot if you want, and be able to pick out a single cell. I'm just going to zoom out of the spatial map because it's going to be really quick, and it actually places a crosshair bang center of the cell that is in wherever that cell is. So that's one way of correlating. So that's why we kept it in that way. Can you draw an ROI and be only the cells in that ROI in a UMAP? Yes. So with this version, the answer is partly yes. Our newer version will have where you can actually draw an ROI and visualize that on ROI on a UMAP. Let me try to answer that one more time. So suppose I have a custom cell group. So I have drawn, I'm going to try and clear this out real quick. So I have drawn, so as you can see now, if I just have the custom cell group that I have drawn, whichever that is, Yeah, so that's group one. So basically now the group one is being thrown around, all displayed on the UMAP cluster as well. So all those cells are now being displayed on the scatter plot as well. The Vizgen post-processing tool can be found at the GitHub. So here is the post-processing tool. I will paste the link in the chat, or you can simply Google Vizgen post-processing GitHub and you can access that. Could the file be analyzed? Yes. Great question, Bing. So the file analyzed by VPT can be visualized by the software. We have commands that are you can insert in the format there to display and recreate the VZG file so that you can re-import it back into the visualizer. Where can you find? Okay, so I think I have that. Select custom cell groups based on predefined analysis parameters, such as that. Yeah, so that is a workflow that is, I'm not completely sure, Maureen, if I have the answer right now, but I will research that a little bit and get back to you. So there are methods that I know that you can actually definitely analyze out of the visualizer, and you can bring that data back in with your annotations and re-import it back. I want to say that I'll have to give you the full path of how to do that, the workflow for that. I do not have that at the moment, but such an analysis are, to the best of my knowledge, possible. VPT to resegment my cells, but the output files did not include a cell boundary. So the cell boundary parquet file, yeah, we need to, maybe we can take this and discuss this offline. Perhaps if you can send me an email, Cameron, and we can discuss this to see why the cell boundary parquet file was not generated after VPT. But there should be a way to do this. No, so for Hangyu, Liu, the fluorescence of the slide itself, GFP or TD tomato, if you have that on the sample, it's a great question, by the way. So we have a method called in a part of our sample preparation, which is the sample clearing, which is a part of the procedure. And what it really does, it really removes that fluorescence from your fluorescent tagged protein. So then downstream, that fluorescence should not be seen. As an additional precaution, Vizgen has manufactured, and we do sell separately, something called a photo bleacher, which is extremely effective in taming the the fluorescence of your protein. So for those users who have such samples where they may have GFP or a fluorescent protein that is expressing, I strongly recommend that you first photo bleach for about 3 hours and then you run it through the thing. So yeah, if you keep the fluorescence, so the fluorescence will be, so to answer your question, Lynn, that's your second question, if you can keep those fluorescence, I mean, the fluorescent tags can be on the slide, but through the sample preparation steps, they will be removed. So your next question will be, I'm anticipating, is like, okay, how do I then look for these proteins? So there are two ways you can do this. One, if you want to look at the protein expression, there are plenty of like GFP primary antibodies available. So you can definitely take advantage of our co-detection. So just like the cell boundary stains I showed here, you can actually co-detect proteins. So if you want to use your primary GFP, which is compatible with RNA, which means that it's purified, it is BSA free, because those things can bring in RNase contamination and damage RNA in the tissue. So if you have a primary antibody that is, and such primary antibodies do exist, you can actually use that to stain your cells and redo. Yes, I think that will a link will be provided at the end of the webinar for this, and hopefully, and thank you for your interest. I hope I was able to cover most of it. The software is accessible for any MERSCOPE user. You are able to access it free of charge. So and you can install it on your Windows or Macintosh platform. Thank you everyone for your fantastic enthusiasm and interest in this. I'm really humbled by the interest in this, and very excited that some of you might actually, if you have not used the MERSCOPE yet, would actually look at this demonstration and then hopefully be interested in knowing more about Vizgen. With that, I will conclude today's presentation. Thank you so much, everyone, and have a great rest of the day and evening wherever you are. Hope to see you again soon on another Vizgen presentation. Thank you. Bye-bye.